Have you ever wondered how scientists manipulate genes to create new traits in organisms? Well, the answer lies in the fascinating field of genetic engineering. This branch of science is a testament to human ingenuity, allowing us to rewrite the genetic code of life itself. Genetic engineering plays a key role in many areas, from medical research to agriculture. It's the powerhouse behind revolutionary breakthroughs like genetically modified crops and gene therapies. But how does it work? Well, there's a whole suite of techniques that scientists use to manipulate DNA. In this series, we'll be exploring a few of these techniques, including the isolation of DNA, creating genomic and cDNA libraries, gel electrophoresis and blotting techniques, DNA hybridization, site-directed mutagenesis, restriction fragment length polymorphism, DNA fingerprinting, and gene synthesis and sequencing methods. So, ready to delve into the heart of genetic engineering? Dive in with us as we unravel the mysteries of genetic engineering. First off, before any manipulation can occur, scientists need to isolate the DNA. You might wonder why this is the case. DNA isolation is the very first step in genetic engineering, because before we can tinker with the genetic code, we need to get our hands on it. Think of DNA isolation as a treasure hunt. The DNA, our treasure, is intricately tucked away inside the cell's nucleus. To reach it, we must first break open the cell, a process known as cell lysis. This is followed by a purification process to remove all the unwanted cellular components, leaving us with a clean, pure sample of DNA. The isolated DNA then serves as a blank canvas for scientists, enabling them to explore, understand, and even manipulate the genetic code. It's like having the keys to the biological kingdom, opening up a world of endless possibilities. With the DNA isolated, we can delve into the real wonders of genetic engineering. The next step involves creating genomic and cDNA libraries. These are collections of DNA fragments representing either the complete set of genes found in an organism or a subset of these genes. In a genomic library, we have a comprehensive representation of the genetic content of a particular organism. It's like a comprehensive bookshelf housing all the genetic information. In contrast, a cDNA library or complementary DNA library only contains the genes that are actively expressed in the cell at a particular time. It's more like a curated collection of the most popular books of the season, if you will. These libraries are created using a host organism, typically a bacterium, which replicates the DNA fragments. In this way, scientists can store, replicate, and analyze large amounts of genetic information. These libraries serve as a valuable resource for scientists in their genetic engineering endeavors, effectively acting as a reference library for the genetic information they are studying. Now that we have our DNA and libraries, we can proceed with gel electrophoresis and blotting techniques. These methods are like the detective tools of genetic engineering, helping to unravel the mysteries of DNA. Gel electrophoresis is a procedure that separates DNA fragments based on their size. DNA samples are loaded into wells of a gel and an electric current is applied. The fragments, behaving like tiny runners in a race, move through the gel at different speeds, with smaller fragments reaching the finish line faster. Following electrophoresis, we move on to blotting techniques, specifically southern blotting. This method transfers DNA fragments from the gel onto a membrane for easier analysis. It's like capturing the runner's footprints to examine later. The DNA is then exposed to a radioactive probe that binds to the specific sequence of interest, providing a snapshot of our runner's path. These techniques allow scientists to analyze the DNA and prepare it for further manipulation. Once we have our DNA analyzed, we can move to DNA hybridization and site-directed mutagenesis. These are not just fancy terms, but key techniques in the world of genetic engineering. DNA hybridization is a process where two strands of DNA from different sources are combined to form a single DNA molecule. It's like a perfect DNA matchmaker. partnering complementary strands to create a new hybrid. This hybrid DNA gives us a unique window into comparing genetic sequences and understanding genetic relationships. On the other hand, site-directed mutagenesis is a bit like a skilled artist, meticulously modifying the DNA at specific locations. It's a precise method that allows us to change the DNA sequence, enabling us to study the function of a specific gene or even create a new trait. It's an essential tool for genetic engineers, as it provides the blueprint for genetic modification. These techniques are integral in modifying the DNA to create new traits. Moving forward, let's explore the restriction fragment length polymorphism, RFLP, and DNA fingerprinting techniques. Restriction fragment length polymorphism, or RFLP for short, is a technique that scientists use to analyze the variability in length of DNA fragments, resulting from restriction enzyme digestion. It's like a molecular scissors, cutting up the DNA at specific sequences. This variability or polymorphism can be used to differentiate between individuals, species, or strains, and is particularly useful in genetic mapping. On the other hand, DNA fingerprinting is a method used to identify an individual from a sample of DNA by looking at unique patterns in their DNA. Much like the swirls and whorls on the tips of your fingers, each individual's DNA holds a unique pattern, making it a powerful tool for identification in forensic science, parentage testing, and genetic research. These techniques provide unique insights into the DNA, allowing for more precise manipulation. Finally, we reach the culmination of genetic engineering techniques with gene synthesis, gene machine, and gene sequencing methods. So, let's dive deeper into these fascinating processes. Gene synthesis is the process where DNA sequences are artificially created in the lab, rather than being isolated from a biological sample. The beauty of this method lies in its ability to construct genes that may not exist in nature, opening up a world of possibilities in genetic engineering. Imagine, if you will, creating a gene that could potentially solve world hunger or cure a lethal disease. That's the power gene synthesis holds. Onto the gene machine, a device that automates the chemical synthesis of oligonucleotides, small pieces of DNA. Picture a machine churning out custom-made sequences of DNA like a 3D printer for genetic material. It's not science fiction, it's science fact. These machines allow for rapid, high-throughput production of specific DNA sequences, speeding up research and development in numerous fields from medicine to agriculture. Now we come to gene sequencing methods, the process that determines the precise order of nucleotides within a DNA molecule. Think of it as reading the genetic code, letter by letter. This knowledge allows us to understand the function of genes, how they interact, and their role in health and disease. There are several methods of gene sequencing, from the classic Sanger method to next-generation sequencing technologies, each with their own strengths and applications. These are the final steps that bring all the previous techniques together and allow for the creation of new traits in organisms. It's like putting together a complex puzzle, each piece a technique, each connection a step closer to understanding and harnessing the power of our genetic material. Just think about the potential applications, from customized treatments for genetic disorders to crops that can withstand harsh conditions. The possibilities are as limitless as our imagination. And that, dear listeners, is the magic of gene synthesis, gene machine, and gene sequencing methods in genetic engineering. In the vast world of genetic engineering, we've only just scratched the surface. Today, we've journeyed through a fascinating array of techniques that scientists use to manipulate, read, and understand the code of life, our DNA. We started with the isolation of DNA, the fundamental first step that enables researchers to work with this molecule of heredity. We then moved on to the creation of genomic and cDNA libraries, collections of DNA fragments that serve as invaluable resources for studying an organism's genetic makeup. Each library represents a different approach to cataloging life's diversity, providing researchers with a wealth of genetic information. We explored gel electrophoresis and blotting techniques, powerful tools that allow scientists to separate and identify DNA fragments based on size and charge. These techniques, when combined, provide a detailed picture of DNA structure and function. Next, we delved into DNA hybridization and site-directed mutagenesis. These techniques allow us to probe the mysteries of DNA, to locate specific gene sequences, and even to introduce targeted mutations for research or therapeutic purposes. We also discovered the fascinating world of restriction fragment length polymorphism, or RFLP, and DNA fingerprinting, powerful techniques that can reveal the unique genetic profile of an individual, a crucial tool in everything from criminal investigations to paternity tests. Finally, we explored the cutting-edge world of gene synthesis, gene machines, and gene sequencing methods. These are the tools that are pushing the boundaries of genetic engineering, allowing us to read, write, and even design our own DNA. Subscribe for more because you won't find me later. Stay curious, keep exploring, and remember, the world of genetic engineering is as vast as our understanding allows it to be. This journey we've embarked on together today is just the beginning. There is so much more to learn, to discover, and to understand. So until next time, keep asking questions and keep pushing the boundaries of what we know about life and its genetic blueprint.